Hi, my name is Kaitlin Marquis. I'm a graduate student in Rick Bushman's lab at the University of Pennsylvania, and I'm going to talk to you today about my project on Hiv replication, transmission, and the metabolome of the female reproductive tract. So we know that women who are colonized by lactobacillus dominant communities, or LDCs, are at a lower risk of acquiring HIV compared to those colonized by highly diverse microbial communities, or HDCs. And metabolites can play a critical role in this process, as it's been shown that lactobacillus-mediated production of lactic acid can directly inactivate HIV virus particles, it can regulate inflammatory cytokine production by vaginal epithelial cells, and it can prevent the growth of pathogenic anaerobes. However, we lack an understanding of how HDC-associated metabolites contribute to this process, mainly due to the inherent polymicrobial nature of the HDC community. So with that said, I want to address this question in the field, and to do that we're going to look at three main topics today. The first is, can we identify conserved changes in the HDC versus LDC metabolome? 2, could any of those changes affect HIV replication in target cells? And 3, what's driving those changes in the metabolome? Is it differences in microbial metabolic machinery, host response to colonization, or both? So we started out by asking what are some conserved changes in the HDC versus LDC metabolome. And to do this, we conducted a meta-analysis on metabolomic studies that collected vaginal fluid from either HDC or LDC colonized women, and then we calculated the relative fold change in metabolite concentration across these studies. The results are shown in this heat map where every row is a different metabolomic study and every column is a different metabolite that significantly and consistently changed in at least 3 out of the 4 studies. And of interest, most of these changes are in amino acid metabolism, possibly suggesting to us that there might be a higher reliance upon amino acid catabolism in women colonized by HDCs. So now that we know that there are clear changes in the metabolome, we wanted to address whether or not any of these could affect HIV replication. So to answer this question, we screened a large library of over 500 metabolites to identify putative enhancers and inhibitors of HIV infection. So here's the workflow of the screen. We use cells that express the HIV receptors and pre-treat them with metabolites for 24 hours before infecting with a dual-tropic HIV. Then 48 hours later, we can conduct a luciferase assay as a proxy to measure virus replication. So if we overlay the results from the high-throughput screen with data from the published metabolomic studies, we can identify a small subset of cervicovaginal metabolites of interest, which is what I'm showing you here in this heat map. So we have particularly focused on this 2-hydroxyisovalerate, or 2-HV molecule, as this metabolite was enriched in HDC fluid across all 4 studies, and it increases HIV replication in our TZMBL screen at high concentrations. So we then set about to validate our screening results by measuring the effect of 2-HV on HIV replication in primary CD4 T cells using 2 different methods. On the left, I'm showing you the results of an ELISA-based assay where we measure the amount of HIV P24 protein in the supernatant 7 days post-infection. So here you can clearly see that there is a dose-dependent increase in the amount of HIV P24 protein in the supernatant in cells that were treated with 2-HV. And this was analogous to the effects that we see with treatment with our sodium butyrate positive control. And this was also supported by flow cytometry data from 5 different donors that were treated with 2-HV and then infected with an R5 tropic HIV strain. So across donors, we can see that there's a 3 to 6-fold boost in the amount of HIV-infected cells after treatment with 2-HV, and this is again comparable to treatment with our sodium butyrate positive control. So naturally, this leads us to the question of what is the mechanism for 2-HV-mediated boost in infection. Now we know from data from Doug Kwan's lab that there are more activated HIV target cells in HDC fluid. This is characterized by CCR5 CD38 HLA-DR triple positive cells. So we measured this cell population using flow cytometry in 2-HV treated cells in both a virus-infected and a mock-infected background. However, we did not see any consistent pattern of remodeling, suggesting that 2-HV treatment may be directly affecting a specific stage of the HIV replication life cycle rather than just blanket T-cell phenotype. So the last question I want to address is basically what is the source of this elevated 2HV levels in HDC fluid? So we asked the question whether or not specific vaginal microbes can secrete 2HV. So we basically just grew up a bunch of representative HDC and LDC strains that are associated with either HIV protection or acquisition, and then conducted mass spectrometry on the supernatant. And what we've identified so far is that at least one HDC strain, so this Veillonella montpellierensis, can secrete high levels up to 30 nanogram per microliter of 2HV into the supernatant. Unfortunately, at this point, we're not able to predict the ability of different strains to secrete this metabolite based solely upon genetics. So you can see here on the left is the predicted synthesis pathway for 2HV, and on the right above each of these bacteria, I've broken down into whether or not they have those enzyme homologs in the genome. And so what's obvious is that while all three HDC strains theoretically possess the machinery to make 2HV, in practice it's not being secreted by all strains. So this is something that we're currently trying to follow up on and understand in the lab. So in summary, what we've identified so far is that there's at least one HDC strain that can secrete 2HV into a supernatant, and we know that this metabolite can boost HIV infection in primary CD4 T cells. We're currently determining whether or not there are also more 2HV metabolite precursors secreted by HDCs, and whether those can similarly boost infection in T cells. And we also need to determine at which step of the HIV life cycle that 2HV affects, and whether treatment affects vaginal epithelial cytokine production and immune cell chemotaxis, as that has biologically relevant implications for transmission. With that, I'd like to thank you all for listening, the entire Bushman Lab, especially Rick, my thesis committee members, our collaborators, and to those organizations that have provided us with funding and reagents. So thank you for listening, and I'm happy to take your questions.